You know, let's just talk about just general bacterial properties today, and over the next week or so, we'll talk about properties of bacteria that allow them to actually cause infection. We won't have another example. And then we'll actually get into the fun stuff. We got all this background stuff. And then we'll start talking about actual diseases, which is, why we're probably here. Okay. So, for those of you that had Gen micro, which is how many people had Gen micro in here? Everybody, right? Did anybody have basic micros pre-rec? Okay. Basic micro used to be a pre-rec for this course. I think they kind of changed that in another way. Ultimately, so you all know this, so I'm probably not going to spend too much time on this stuff. Bacteria come in different shapes and sizes. If they're rod-shaped bacteria, the singular is bacillus. If you have one rod, if you have multiple rods, it's bacilli. And then you have Vibrio, which are condom-shaped, kind of boomerang-shaped bacteria. So you see that kind of down here in the bottom middle. We'll go back to this little guy. Spirochetes, so there's going to be corkscrew shape, cocci are going to be round, coccus, singular, cocci, plural. Different things you've heard of that are rod shaped. Actually, since y'all had Gen micro, I'm not going to go that far back. You've given some examples of rod shaped bacteria. Yeah, E. coli, bacillus, things like that, salmonella. For the cocci, what about some examples of cocci? Those are three of these are just simple names. Let's go rod. So rod shaped, so streptococci, staphylococci, amylococcus. For a lot of cocci, not all, but a lot of it has coccus in the genus name. Vibrio is a super easy one. So we give you an example of Vibrio. Yeah, it's a name, sort of like, yeah. Vibrio, cholera, cholera, things like that. What about spirochetes are a little bit more difficult because we generally don't talk about as many of those, at least as they cause like human disease because there's not a lot of them. Anybody remember any spirochetes?
Bacteria?
That's a rock.
Okay, never mind.
That's all right. There's not many.
Lyme disease?
Lyme disease instead of the Lyme. Did anybody remember what the what? Cholera is Vibrio cholera. That's kind of a boomerang shape.
Isn't it Bacterial?
Good. Bacterial dorferi. It's named, the species name is named after Willie Bergdorfer. Willie passed away here 5 years back or so. He was the one that actually found the vector for Lyme disease, not the bacteria itself, but they named it after. So Bacterial dorferi is an example of a spirochete. There's one other one we talk about. Causes sexually transmitted infection. But STD, STI. Syphilis, good. You remember what the conservation of syphilis is? I know I'm really making you be here. We talk about that in Gen micro, but it's not in depth. Do you remember? Streptococcus causes syphilis. Those are literally the only two spirochetes we generally talk about. Borrelia and Streptococcus. When we talk about spirochetes, remember they're a little bit unique. They make flagella, but they make endoflagella. Sometimes they're referred to as axial filaments. So instead of the flagella, we'll talk a little bit more about that a little bit too. Remember, flagella extend to the surface of these bacteria. They rotate. When they rotate like a reverse group that allows them to move forward. So it's typically flagella are outside the bacterial cell. And spirochetes is a little bit different. They're actually inside that periplasmic space. So they're inside between the inner and outer membrane. Let's do another question. I'm going to really test your data to see how much you remember from Gen micro. So if it has an inner and an outer membrane, it's either gram-negative or gram-positive. And I'm going to tell you what people are going to tell you the truth. Either gram-negative or gram-positive. Gram-negative. The thing about these spirochetes is you would think based on the structure, but the outer and inner membrane is gram-negative. Actually, distantly related to gram-negatives and gram-positives, or not even really gram-negatives. They also don't stain with the crap. Reason being is they're very thin, so that image up there, that spirochete, the diameter of or the width of the cell is very, very thin. They might stain like a gram negative, but they're so thin you're not going to see them. It's very, very thin. So they make the flagella. What happens to those flagella in the periplasmic space, or the actual filaments, is they move past one another. So when they kind of move past one another, they cause a torque that makes that corkscrew actually turn. So when that corkscrew turns, it pours down into your genital cells to establish an infection. And we're talking about infections. So that's what essentially those exofilaments allow to attach and invade it. Spirochetes are kind of weird. We'll talk about tryptamine a little bit, about STIs. We talk about Boreal in the spring. Those are pretty much similar. There's another one we talk about a little bit in the spring, but those are pretty much the main two we'll talk about for spirochetes. There is another one that's not out there. So they're also Spirillum. So what a Spirillum is instead of the periodicity of this corkscrew looking like this, like a spring, it's more stretched out. So it looks more like I want to turn off the lights so you're going to have a really hard time seeing this stuff today. I might have to turn them off anyway. So they look more like this. See how the corkscrew is not as tight? If you look at this, it looks like a seagull kind of in flight. That's a specific kind of bacteria. We're looking at the gram stain, what it is. You have that gull wing shape on the gram stain because if somebody has like some kind of GI symptoms, that's Campylobacter, has that gull wing structure. Campylobacter and Helicobacter are pretty much the only ones we'll talk about that are Spirillum. Campylobacter causes GI issues. We'll talk about that in the spring semester. And so Helicobacter, anybody know what Helicobacter model, right? Yes, that's awesome. So not a lot of examples of spirochetes and Spirillum, but there's some. And the reason all this stuff matters is if you have somebody that comes in with symptoms, you take a patient sample and then you do a gram stain from the micro lab. That's going to impact what treatment you decide to use. So you're going to treat somebody that has an infection with a gram negative ron a lot differently than how you treat somebody that's infected with a gram positive toxin. So you usually use gram stains early on to impact what empiric therapy you're going to use. I realize you haven't talked about this probably anything else. Some of you had varieties, you don't remember some of this. Anybody remember what the difference between empiric therapy and targeted therapy is? So you have somebody that comes in and I'm just trying to think of it's a random infection. It doesn't even matter. They come in with a random infection, you know it's bacterial, right? Or I should say no. You're pretty certain it's bacterial. So you prescribe them an antibiotic. We haven't got lab results back yet, right? Because it takes time to grow this stuff up. And you don't know what it is necessarily. You might have a good idea, but you don't know for certain. So you put them on empiric therapy. So empiric therapy means you're trying to give them a broad spectrum antibiotic that's going to cover everything that you think that they might have. So you have, usually when somebody comes in, you have a differential diagnosis. So you have, say, 10 things you think might be causing this infection. The most likely ones are towards the top, and then the really unlikely ones are towards the bottom. If somebody comes in with some kind of bacterial pneumonia, where you're going to have things high on the list that might be things like staph or whatever. But then on the bottom list, you might have respiratory anthrax. Chances are, but probably not respiratory anthrax. But they could have respiratory anthrax. Just very unlikely. So that's how you do differential diagnosis. So if you look at that list of things, you try to look at the most likely things and find an antibiotic that's going to wipe all those things out. That's your broad spectrum antibiotics, there's a chance that there might be a bacteria in that population, a bacteria that's causing infection. If it has one mutation, some kind of mutation, becomes resistant to that antibiotic. And when you remove the antibiotic from its broad spectrum, those bacteria are going to grow. And then you can use that antibiotic to treat it again. So you essentially took something that's like nukes everything off your list because you can't use it anymore because bacteria is resistant to it. So you want to use targeted therapy. That makes sense. Good. Okay. We'll talk about that a little bit as we kind of go. I won't talk about therapeutics in here, but I do want to make sure you understand kind of why we're doing this stuff. Like the shape of the gram and all that stuff does matter. This is the arrangement, I think we have talked about. Yeah, I didn't talk about 10 micro, but I don't know if you don't remember how long we had 10 micro. The arrangement is dependent upon how the bacterial cell divides, so how it goes binary fission. The bacteria can grow in pairs. They grow in pairs. You put diplo in front of whatever the shape of the bacteria is. So if I say diplococci, you have two sterile bacteria. They're together. There's some bacteria that like that. We'll come back and talk about those a little later. If it's in a chain, streptin. If it's in a cluster, staph. If it's in like groups of 4, it's called tetrad. You don't even know that. But these different arrangements have different meanings. Some bacteria are pleomorphic. Pleomorphic means that depending on what the environment is, they change the shape. So that's why I said I come back to this little guy. So this little guy usually exists more as like a rod, but depending on what environment it's in, it might become longer. This is kind of how Haemophilus influenza, we'll talk about Haemophilus here over the next few weeks. This is how Haemophilus influenza can sometimes look in the lungs. This is actually also how Acinetobacter can look in the lungs sometimes too. Instead of making these kind of rod-shaped bacteria like you would see if you grow on a plate, you grow in the lung, it presses a mucin in the lung, and make these long spaghetti-shaped looking things. Depending on the environment, carbon sources, and its metabolism, it's going to look different. So if you took like a lavage from somebody's lungs, and you got something like this back, it's like, well, that must not be like Acinetobacter because it's not rod-shaped. It could be Acinetobacter because it grows differently when you grow in the lungs. And so that's what pleomorphic means, change of shape depending on the environment it's in. Obviously, you won't know this from Gen Micro. You don't remember this from Gen Micro. You punch myself in the face. Bacteria are small. So you all know that. I remember this as well. Different pieces and parts of bacteria. And we'll walk through all these different parts. But some of them have a capsule. We talked about capsule a little bit in here before. Capsule is going to be this polysaccharide substances on it, carbohydrate-based substances on the outside of the bacterial cells. Not all bacteria make this, some bacteria do. And it does a bunch of different things. So it can help keep it hydrated because sugar's going to absorb water. Also, the sugar water becomes sticky, so it can be used for adherence. And it can also be used to help evade phagocytes. So it makes it very hard if you take a bacterial cell, let's say it's this big, it has this big capsule around it that's this big, makes the size of the bacterial cell a lot bigger, it makes it harder for macrophages to come around and wrap the pseudopods around it than call it. Even if the bacteria, even if the macrophage can take up the bacterial cell, sometimes the capsule helps it evade destruction of metabolites as we talked about on Monday. So capsules do different things. We'll talk about that a little bit more here today too. Cytoplasm, you know what cytoplasm is. That's the fluid inside the cell. That's where all the stuff is. Including ribosomes, including proteins are made. The genetic material, which is not going to be membrane bound, so bacteria don't have a nucleus, except a couple oddball environmental isolates that we won't talk about in here. So for all intents and purposes in here, bacteria, no nucleus. They do condense their nucleic acids, their DNA, in what's referred to as a nucleoid, so it can actually fit inside the bacterial cell. Some bacteria have I'll actually do that, so I can do a little bit of talking. What's a plasma? What's up, Peter?
A piece of genome that's extra.
So it's use the word genome lightly because they can get rid of the plasmid, so it's not a core component of the genome. So ancillary uses certain DNA. What's the plasmid using now?
What pesticides do we have?
Yeah, like orphan genes.
Oh, yeah.
And virulence, antibiotic resistance, which depending on how much of a period you are, that's virulence gene too. So there are things that are accessory or ancillary. Things that the bacterial cell don't necessarily need to just grow on like a petri dish on a petri plate. But they can use for certain environments that can cause infection. It's usually virulence factors, villus genes, antibiotic resistance genes, things like that. Not necessarily for them to survive. Some bacteria have more than one plasmid. Some bacteria have no plasmids. Flagella, we'll talk a little bit more about, but remember those proteinaceous and protein-based components that are outside of some bacterial cells. Bacterial cells have different reasons for this. Some bacterial cells don't make them at all. We'll talk a little bit more about that too. What else? Does everybody remember what the cell wall made out of bacteria? What's the cell wall made out of? Peptidoglycan, N-acetylglucosamine, and N-acetyluronic acid cross-linked with tetrapeptide that's put in there with something called a transpeptidase because we're super original in the way we name crap in microbiology. Sometimes that helps you all. That's good. There are a lot of antibiotics that target things like the cell wall, like pesticides. Those are going to target, we'll talk a little bit about that here in a little bit, but those are going to target cell wall synthesis. When do you think you see in those cell wall synthesis during the stage of growth? Remember we talked about that in Gen Micro too. There was a lot you didn't have for Gen Micro, so you didn't talk about this in Gen Micro. I don't know what the hell you talked about in Gen Micro, but if you did talk about this in Gen Micro, please let me know because I'd be curious of that. We'll see. What's the phases of growth? I'm tired of giving you all the answers. Have a conversation with me. What's the phases of growth? Remember like growth curve? Lag. Yep.
There's a one phase.
Log, so log, exponential, that's all the same thing. And then what? Stationary. And then the last one, it's not all bacteria, but some bacteria can go into death phase and deserve to die on. So those are your 4 phases. So if you have an antibiotic that targets cell wall synthesis, where do you think that's likely happening? Which one is more? During. You can start to see some during lag, but mostly during log. So in theory, what that means, especially if you're talking about this example where you're going to target cell wall synthesis, the best time to give an antibiotic would be while bacteria is in exponential phase, while they're in log phase. That sounds great on paper. The problem with that is if you have somebody that has an infection, there's going to be bacteria in that population that are, some are going to be stationary, some are going to be in log, some are going to be in lag. In practice, it doesn't work. But if you're looking at it in a controlled system, like in a broth culture in a lab, in a closed system, they would be most susceptible during log phase. Okay, we'll come back to that in a little bit. Plasma membrane, you want to know what that is. We'll talk about the gram-stained mailing here. Fimbriae, we'll talk about those as well. So technically, fimbriae are short protein projections coming off the surface of a bacterial cell, and there's a lot of them. If you're talking about pili, they are usually fewer and far between, and they're a lot longer. The problem with that is, and you'll see this as we talk about some of these bacteria here throughout the semester, is we have interchanged those names so much that they don't really mean that anymore. But technically, fimbriae are supposed to be a ton of them, and they're short. Pili is supposed to be like 10 to 20 of them, and they're supposed to be really long, is the technical definition. We've looked that up over the past 20 years somehow. What else? Inclusions, we'll talk about some examples of inclusion bodies. Those are kind of cool. They tend to be more for environmental isolates, but we'll talk about it a little bit. Mesosomes, don't worry about that. Sometimes you see bacteria, if you do electron microscopy, they have like these little like structures and vaginations and things on the outside of the cell. Those are usually fixation errors. So it's just a process of preparing the bacteria to look at it under the electron microscope, causes cell. They're not really structures, it's just moved to, because that's the only way we can find the protein. Again, nucleus in here, we're going to talk about this, nucleus. But the bacteria, the bacterial DNA is compacted into something called a nucleoid. Sometimes you see people refer to that as nuclear body. Most people are going to use the word nucleoid. It doesn't have a nuclear membrane, doesn't contain a mitotic apparatus, so you're not going through mitosis. You get the bacterial cell. You go through the process called binary fission. Binary fission, essentially you have two bacterials, or you have one bacterial cell, and that one bacterial cell divides into two bacterial cells. You have this protein ring, which is called FTSC. You don't even know this is where this stuff found. The FTSC is going to contract out like a belt, and when that contracts, it'll make two new cells. So it's not mitosis. FTSC is a homologopactin. All stuff you would learn in a microphysiology course, but it's likely at the graduate level stuff you don't even know yet. In the nucleoid, you have polyamine, magnesium ions, then you have DNA, obviously, the supercoiled, double-stranded, RNA polymerase, protein, blah, 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 blah. Cool thing about bacteria, and you probably don't talk about this in Gen Micro too, is, well, actually, so in our cells, you have DNA and nucleus, right? But you can't carry out DNA synthesis. I don't know where it is. Where are proteins made in our cells? Outside the nucleus, right? It takes us a lot longer to make a new cell than a lot of these bacterial cells. Oftentimes. These bacterial cells can undergo DNA synthesis, transcription, and translation all at the same time. So as you're transcribing RNA from DNA, that new piece of mRNA that's being transcribed can already have a ribosome on it, can be making protein while it's being made. So all this happens at the same time. Because the things aren't I don't say reactions, but the reactions aren't compartmentalized because there's no nucleus. Everything's inside the plasma, it's all together. So if you can do all that stuff at the same time, those cells can divide a lot faster. And some of these things, like some of the E. coli you've got, divides over about 15 minutes. I mean, they move. Are they accident prone? Yeah, they can make all kinds of mistakes. But a lot of those mistakes are going to mean the bacterial cell dies. Unfortunately, a lot of those mistakes means those bacteria might get a point mutation, and that point mutation might make it resistant to an antibiotic or something. You don't know. So it's kind of good and bad. Every time you have a new bacterial cell, the bacterial cell that's made is going to be a clone of the previous cells, and the genetic material, the genetic sequence should be exactly the same. The only way they really get diversity, and like genetic diversity, is through mutations and some other things we'll talk about here towards the end of today, like conjugation, transduction, and all that stuff. Cytoplasm, again, no organelles. There could be storage granules. The ribosomes are 70S, our ribosomes are 80S. 70S ribosomes in bacterial cells are made up of a 50S and a 30S component. I know 50 plus 30 is not 70. And our 80S ribosomes are made up of 60S and 40S components. What this means, if you have an antibiotic that's going to target a bacterial cell, it's preferable that it's targeting something the bacterial cell has that you do not have. So ribosomes are a great way to do that because your ribosomes are 80S and theirs are 70. So you can target that without any side effects. When you start to get into some of the antibiotics like the polymyxins, like D and E and stuff like that target gram-negative cells, it targets gram-negative cell membrane. You obviously have cell membranes. There's not that much difference between phospholipids and phospholipid. Cell membranes is cell membrane. Your cell membranes aren't a ton different than bacterial cell membranes. The components of them, like the ratios of the different phospholipids are different. They're very different. They're still phospholipids, they're not bad. So you start to see side effects of those kinds of things. Polymyxins, daptomycins, things like that. For the ribosomes, drugs like tetracycline, aminoglycosides are going to target ribosomes of bacteria. So that's going to make protein synthesis. There's bacteria, you can protein synthesis, bacteria can't make new bacterial cells. Cytoplasmic membrane, again, the plasma membranes and plasma membranes, permeable. Cytochromes and enzymes involved in electron transport on the inner membrane of bacterial cells. Also oxidative phosphorylation, which means making ATP. All kinds of carrier lipids and enzymes, penicillin binding proteins involved in cell wall biosynthesis. So one thing I will tell you, because this might be a little confusing later on to you, but I'll explain to you now. When the process by which cell walls made bacteria was originally outlined, a lot of those experiments involved using penicillin. Penicillin blocks cell wall synthesis. So a lot of the proteins involved in cell wall synthesis are named penicillin binding proteins because they bind penicillin. Obviously, that's not what they're normally used for. They're used to make new cell wall. But a lot of those proteins that make cell wall are going to be called PEP proteins. They're going to be penicillin binding proteins. That's all that is. Enzymes involved in phosphorylation, DNA replication, cytoplasmic membranes and lipid proteins, and some carbohydrates. And again, there's certain antibiotics that work on plasma membranes. We haven't talked a lot about it, but polymyxin, staphylococcus, that's pretty much the one. And there's a lot of side effects with those usually too, because again, you can get plasma membranes too. Kind of what it looks like, cytoenergy reductions, cytoplasmic membranes, that's where ATP is made. That'll come into play again a little bit, I'll explain that to you. Remember when bacterial cells go through the electron transport chain, they're going to pump protons out. In this example, in the periplasmic space, when those, so proton motor force is set up enough, protons come back in to the cell, that can be used to do different things, like spin that flagella to make the bacteria. There's membrane proteins associated with plasma membranes, some of them are peripheral, some of them are integral, which just means that they're attached to one leafless as plasma membrane for peripheral, versus integral means they go all the way through the membrane, they're embedded in that plasma membrane. And those integral ones are the majority of the membrane proteins that exist in bacterial cells and are used for these things, transport, secretion, energy conservation. Difference between the bacterial cell wall. Gram-positives, remember, have a very thick peptidoglycan layer. Gram-negative cells have a very thin peptidoglycan layer, so that's what that little schematic shows you there. Gram-negative cells are going to have an inner membrane, and they're also going to have an outer membrane. Gram-positives only have the inner membrane, they don't have the outer membrane. You can use a gram stain, which we'll walk through here in a little bit, to show you which ones are which. We'll talk about that, we've done that in just micro too. The bacterial cell wall maintains rigidity and shape. Protects from osmotic shock, responsible for imparting antigenicity to an extent, important for variance and immunity, and again, determines that gram reaction. So remember, for those of you that haven't for Gen Micro, that peptidoglycan layer is going to function kind of like a chain link fence, so it's going to have some play to it, it's going to have some plasticity to it. So if you take a bacterial cell and you grow it into 100% water, that peptidoglycan is going to have a little bit of flexibility to it. It's going to allow that cell to expand and contract a little bit so it doesn't lyse from osmotic shock, from osmotic stress. If you did that, if you drew like one of our red blood cells in water, it's going to burst because of the osmolarity differences between the inside of that red blood cell and the water you put it in. So that's what the cell wall is used for. If you were to say, have a bacterial cell that did not make peptidoglycan, that did not make a cell wall, what shape do you think it would be? If you even give it even a try.
Random?
Huh?
Random? Circular?
Certainly. So think about vinegar oil that makes salad dressing. If you shake it up, all the little lipid droplets are spherical, right? That's the sphere is the lowest free energy, or the lowest G. That's the simplest shape you can get. So like if you took syphilis, like the troponil, like the spirochete, and you got rid of genes involved in making cell wall, and there's also a bunch of cytoskeletal genes in there. If you get rid of genes involved in making that the cytoskeletal components inside of the human, it wouldn't be a sphere. There's a lot of things. The more complex the shape, the more cytoskeletal components you need to actually make that shape. So spiral feature is complex. There's treatment positives and gram negatives, not all of them, but most of them. Underground peptide glycan. There are a few examples we'll talk about in here, not many that don't make a lot of peptide glycan, if any. We'll come back to those. Gram positives make techoic, lipotechoic, telluric acids. Essentially what this means is anytime you see TEICH, it's a gram positive. They love asking these kind of things on board exams and MCAT, things like that. If you see anything where it's talking about tech anything, it's a gram positive. That's a nice way to grow them fast. And they make polysaccharides, gram negatives again, they make peptide glycan, they make lipoprotein, and they have that outer membrane. The outer membrane has lipopolysaccharide in it, and lipopolysaccharide we'll come back to here in a moment, has a component called lipid A. Lipid A is an endotoxin, so it can cause septic shock if the bacterial cell slice. So this is an example we'll talk about as we go throughout the semester a little bit. Unfortunately, this is usually one of the more common examples which I give it. You have somebody that comes into a nursing home, they're there, they end up getting put on a urinary catheter. The staff is monitoring a lot of patients in the nursing home. They don't pay as much attention to that urinary catheter as they should. The person gets a urinary tract infection, it goes missed, nobody notices it. The urinary tract infection, said UTI, so many words all the time. The UTI gets worse, and it proceeds into urosepsis, which means the bacteria get out of the urinary tract into the bloodstream, and then you have a bunch of gram-negative bacteria in an elderly patient's bloodstream. After the nursing home has missed this for about 2 weeks, they suddenly realize that this patient's not doing very well, and they rush them into the emergency department, and the emergency department is like, this person's on septic with like E. coli. You give them antibiotics, and they die. You give them antibiotics, you have to give them antibiotics, but you give them antibiotics, it's going to lyse all those gram-negative cells. That when you lyse those gram-negative cells, it releases lipid A, and you go into septic shock. The problem with septic shock is it's very hard to treat because you don't know how the patient is going to present with that septic shock. Some people have increased like vasodilation and like leaky vasculature where they're leaking out, essentially like they're bleeding out. Other people get a lot of clots. Some people get kind of both at the same time. So you would think it's like if they're clotting, you treat them with a blood thinner, but you can't really treat them with a blood thinner because they might bleed out. You can't do that. You go into something called DIC a lot of times. So you can't. You're trying to treat like opposite symptoms in the same patient at the same time. So if that infection gets bad enough, the sepsis gets bad enough, unfortunately there's not a lot you can do sometimes. And that unfortunately seems to be one of the examples I've seen more often. Usually patients elderly, nursing homes just have too many patients to look after, they miss it, and by the time they get there, there's not a lot you can do. Difference between a gram positive and a gram negative. This is what it looks like. So can anybody tell me what kind of electron micrograph this is? So you're doing, well, let me bleed in here a little bit. So you're doing cross sections. So you're doing electron microscopy, you're doing cross sections. So you're taking a bacterial cell and you're cutting it into slices. So you can see the cell membrane. You can see that little thin line there's peptide glycan. This is gram negative. We'll come back to this little guy. But outer membrane, periplasmic space, this little thin, like thread-looking line is the peptide glycan, and then this darker line here is the cytoplasmic membrane or inner membrane, or plasma membrane, it's all the same thing. What kind of electron? Which one?
SEM.
So SEM you use with the surface usually. So T is a transmission electron microscopy you use to do sections. Typically, not always, you can get away with some different methods, and that's not usually. You can pull some tricks, but this is TEM. Down here, we might talk about these on occasion, especially in gram negatives. You don't see, and I'll ask you this in a little bit. These gram negatives can make what's referred to as an outer membrane vesicle. And packaged into those little outer membrane vesicles can be things like virulent factors. They can let these outer membrane vesicles off. They can come into contact with our host cells and release the contents of those outer membrane vesicles to our cells and cause damage. Not all gram negatives do that, but some do. So why don't you see this on this guy?
Does that matter?
Okay.
Okay.
So outer membrane vesicles, gram negative, you see this little blood. Why don't you see outer membrane vesicles over here in gram positive? Because gram positives don't make it out. They don't have an outer membrane. There's nothing to blood. A little cartoon, little diagram here, what gram positive looks like. Super duper thick peptidoglycan. Gram negative, super duper thin peptidoglycan. And also you have this little polysaccharide. Peptidoglycan, again, both gram positive, gram negatives are going to have it. 50% of the cell wall and gram negatives are peptidoglycan, but only 2 to 10% of the gram negatives. It's found in pretty much all bacterial cell walls except mycoplasma, mycoplasma, we'll talk a little bit about when we talk about respiratory tract infections. It's kind of not all. Depending on how old the textbook is you read, you might see where mycoplasma doesn't make any peptidoglycan. There's been some studies done where you can detect a little bit of it, but it is a infinitesimally small amount of peptidoglycan, not enough to make like a real cell wall. Again, the peptidoglycan is made up of those two compounds down there, N-acetylglucosamine and N-acetyluranic acid, cross-linked with a tetrapeptide that is inserted between N-acetylglucosamine and N-acetyluranic acid with an enzyme called a tetrapeptidase. That reaction itself is the target of a lot of different antibiotics. They're going to target cell wall synthesis. So that means peptidase is a target for some antibiotics. This is what it looks like. You don't really need to know the intricacies of this. Just know this is those two compounds are cross-linked with the tetrapeptide. You don't need to know the tetrapeptide is made out of alanine and lysine. Okay. Is there anyone around to understand? I see 3 heads popping, yes. Okay. So let's walk through how to do Ramsey. I usually do this on the board, but I might do it on the slide today for these tracks. Okay. So, what slide am I on? No, we're halfway there. Cool. Just want to make sure I don't lose track of time. So you have a specimen that you want to do a gram stain on. You take the bacterial cells, you put them on a slide, usually mix that with a little drop of water, and then you usually do something called fixation. So there's 2 different ways you can do that. You take the slide, you can pass it back and forth over like a flame real fast. You don't let it handle this flame for too long or else you'll get plastic with glass particles everywhere. I've seen that happen before. Don't do that. Or you can use something like methanol. I'm just going to fix it to the slide, which is usually what we do here in the there's standard jars over there, methanol, you take the slide, you sample after you let it air dry. If you don't let the sample air dry the slide first, you just put it directly into the methanol, or just out into the methanol, let it air dry, stick it down in there. That's going to do a couple things. It's going to fix the things to the slide. And it's also going to kill the bacterial cells. If you take that bacteria then after you fixed it, and you add a stain to it called crystal violet. So crystal violet is considered a primary stain. It's the first stain you have. That's why it's called a primary stain. So you flood it with crystal violet. This is the way we teach that in gym microenvironment in the lab. It tells you to do that without them. The more you do, the easier it is that time because you get used to doing it. You can do it in 15 seconds if you don't want to. We usually do kind of the fall this minute. So you let it sit on there for a minute. Then you add iodine into that drop of crystal violet. Your lab notebook says to rinse off all the rinse off all the crystal violet, then add iodine. That makes no sense. You want the iodine and crystal violet to be in contact with one another. because the iodine serves as a mordant. So what a mordant does is the iodine binds onto the crystal violet and makes big complexes of crystal violet dye and iodine. If there's no crystal violet there, because you press it all the way for the iodine to bind onto, you're not going to get a lot of big complexes because all the crystal violet got washed away. So you do that for about a minute as well. And then you drip over alcohol over top of your specimen for about, it's not necessarily a time. I like to tell students you drop it over the slide until you start to see the droplets coming off the slide turning clear. It's usually a good way to do it. If you do it too long, you can over colorize, which means you take the stain out, even out the cells you want to be in. Then you rinse with some water to get rid of the residual like staining crystals and alcohol. And then you do a second stain, which is saffron. And the saffron, you let it sit on the sample for about a minute. And then you rinse out on some water. I will walk through this again on the board because I realize why I usually walk through it on the board. As you go through that process, gram positives that have the thick cell wall will be purple and gram negatives should be green. There are some ways we can do this up. You can colorize, which means you use too much alcohol and you start a purple stain out of the gram positives. There's things you can do, but not too hard of a procedure. So let's do it like this.
There's a marker on the other side.
Is there? Oh, but it's purple too. Perfect. Okay. So, let's walk through this. So it makes sense. So you have this rod-shaped bacterial cell. Let's just say it's rod. Rod-shaped bacterial cell. You add the crystal violet. Crystal violet is going to go inside these bacterial cells, whether it's a gram positive or gram negative. So at this point, everything's going to be purple, right? So everything's going to be purple. Then you add your iodine. Everything's still going to be purple, right? So then you add your alcohol. Okay. So that's the point I wanted to get back to. So when you add your alcohol, this is your differential step. This is when you, in theory, can tell the difference between your gram positives and gram negatives. What happens? Steps 1 and 2, you've made these big complexes of crystal violet and iodine. You get the drift. Big complexes of crystal violet and iodine. The cell wall for gram positives is so thick, it's going to trap this big guy inside of the cell and it won't be able to get out. Gram negatives, thinner peptide glycan layer. I want to hammer this laser to stay on. Thinner peptide glycan layer, this can get out. The other thing, and depending, I don't know if your lab man tells you this or not, you don't really need to know as much about this, but the phospholipids in gram negatives are different than the phospholipids in gram positives. A lot more of the phospholipids, when you expose them in gram negatives to alcohol, the phospholipids are extracted, so it makes a bunch of holes in the membranes too, that allows that crystal violet iodine to escape a little bit easier. So it's actually two things, but the main difference is gram positives thick peptide glycan, gram negatives thin peptide glycan. So at this point, if you're looking at a sample, this is considered the differential step, because at this point, if you're looking underneath the microscope, if you see a bunch of purple cells, it should be your gram positives. If you look at it and you're like, I don't see anything at all, there's nothing here, but I know I put something on the slide, then you probably have a bunch of gram negatives you haven't counter stained yet. So primary staining, mordant,
is your iodine. Primary stain. Ethanol is your decolorizer. This is decolorizing, right? Everything's clear. I'll make this a step 4, but it doesn't really need to be. Rinse with some water. And then you add the saffron. So the saffron is a counter stain. You also see people call this a secondary stain because it's the second stain in the gram stain. The saffron, if you have, let's do this. So your gram positives will still be purple here, but your gram negatives will be clear. So if you come down here, let's say this is your positive, this is your negative. This is kind of a pink color, saffron. So your gram negatives the clear ones will stain pink. Suppose second pink, sorry y'all. And the gram positives are still gonna be purple. So sometimes I'll have some of you all ask me, so it's like, well, doesn't the saffron go inside the gram positives too? Yeah, sure, it absolutely 100% it goes in there, but the purple is so dark purple, you just don't see it. So this is how you tell the difference between gram positive and gram negative. Does that make sense to everybody? Coming back to it? Alright. So that's how you do a gram stain. Gram stain. What are we talking about? Again, this comes into play with treatment. So if you're looking at a sample underneath a microscope, sometimes by doing the gram stain, not only can you kind of use that to figure out what antibiotics you got prescribed like empirically until you get lab results back, but you can also sometimes tell what the bacteria is without even getting the lab results back. Like somebody comes in and they have like GI problems and it looks like this, that's Campylobacter. I mean, I can see that and my vision sucks. I mean, I can be back there. It's just like, there's not that many things that look like that, that present with those kind of like GI symptoms. If you have something that looks like a seagull flying at you and they come in with like a tummy ache, they got a Campylobacter infection. There's just not anything else that looks like that. You still run stuff through the microlab, but you're pretty certain what it is. This, well, I'll ask you this. What's this? Streptococcus dioxide. What's this guy from? Staph dioxide. Yeah, some of these are not hard. And if you have a gram stain that looks like this, and the symptoms correlate with it, it's not that bad. That's a gram positive rod. It's kind of like a fatter gram positive rod. That looks like listeria. Again, depending on the symptoms, that's kind of looks like listeria. This could be like an E. coli or some other gram negative rod. Some of them aren't too hard to figure out. This one up here could be like a Haemophilus. Haemophilus doesn't stain real well, so now when the color's light, you get these pleomorphic stuff where it's like you have some rods, but then you have these stringy looking things too. They're like big long rods. That's kind of what Haemophilus looks like when you take it from a long sample. Yeah.
If the gram stain is more useful for determining a cell wall when it's not they use like differential stain.
You know what?
Like differential stain.
This is a different thing. Methylene blue, a lot of people will use for acid fast. So they're using the counter stain for acid fast. Depends on the symptoms you're looking at, what you're looking for. We'll get there here in a minute. Yeah. We'll get back to that. Yeah, let's come, because we'll get to that in a little bit. We'll talk about acid fast. There's different stains you can use depending on what you think the organism is you're looking for. Because we won't talk about this, we'll talk about this a little bit. You've seen this probably in the vet's office, I'm sure. LC, PD, I never actually use that acronym, so that's what I think about. Lactophenol on blue. So you use that usually if you think it's something that's fungal. Use that to stains the fungal, lactophenol blue. So some people refer to that as LPCB, lactophenol common blue. I can call it acronyms. But use that if you did something that was like fungal infection, because you tried to gram stain on it, that's gonna be a mess. That stains a lot easier, literally you mix it with some activated cotton blue and look at it, it's just like super easy. If you have somebody that comes in with tuberculosis, you have somebody that comes in with tuberculosis, I'll ask you all that too. Mycobacterium tuberculosis. What does mycobacterium tuberculosis have in it? Endospores are bacilli and Clostridia. Mycolic acid. So mycolic acid is made by some bacteria. Mycobacterium, myco is from the mycolic acid. Try to do a gram stain on it, that mycolic acid's this waxy substance. The staining can't get it past that. So you have to do a different kind of staining technique to actually get the bacterial cells to stain. The bacterial cells are clear. You got to stain them with something there, you ain't going to see them. So we'll get to that here in a second. Again, you use this for empiric therapy, a lot of these gram stains. Performed with a laboratory. 15, 20 years ago, some hospitals, it was academic representative, like a teaching hospital, they would let residents do the gram stains. They quickly realized that that was not going well in many places. So they did not let them do that really anywhere that I've heard of anymore. If you get used to doing these kinds of stains, where you do them like a lot of my clinical friends, you do this like everything over and over and over again. It becomes like a muscle reflex. It's just easy. And that's what I told you, like I've seen people do things on there for 10 seconds. I know somebody that's worked like a State Department health for like 30 years. There's like gram stains done, it's like, it's been 45 seconds. How's that even possible? He's used to doing it, and the results are consistent over and over and over again. If you're a resident that's not having that clinical micro lab doing those kinds of tests all the time, and you go and just try to do your 1 gram stain for the month, you're gonna mess that up so bad. And it's not gonna be good. And what they realized about 15, 20 years ago is the resident would come back up and like, yeah, it's a gram negative rod or something. It's just like, and they give them antibiotics. It's like, why is the patient not getting better? Somebody else would go back down and they're like, that was a gram positive rod. That's a completely different thing. So they don't let them touch things. Nobody knows the risks. Truth be told, they really don't let the medical doctors touch many things in clinical microlab. Maybe some people in infectious diseases, like some ID doctors, but that's about it. Alright, so acid fast stain. You have somebody come in and they have a cough. The cough is productive, which means that they're coughing up sputum. The sputum is blood tinged, which means they have blood in it. You notice that they are losing a lot of weight, so it looks like they're wasting. They report that they have night sweats. So they wake up and night sweats. I don't mean like you wake up and it's like you're a little sweaty, like, oh wow, it must be a little hot in here. I mean like you wake up in the morning and the entire mattress is covered. I mean like it is so like you keep that. Those are symptoms of tuberculosis. So if you, especially like in this country, there are other countries where tuberculosis is more endemic. If they come into this country and report those symptoms, you're probably thinking they have tuberculosis. So you would order something called AFB, which is an acid fast bacillus, which I don't know, I'm not familiar with that.
And we have the grams, but we don't have to have the
It's no, it's just different pathogens will sometimes present with different symptoms in animals, and sometimes pathogens are just different pathogens. I'm not as familiar with the animals, but there are different. Malassezia furfur causes up ear infections using antibiotics, but we find that there's like, you see an HIV patients, they cause these type of pigmentation on their back and stuff. cells AFB. Means you think they have TB. So they send it down to the lab. People in the lab know not to do a gram stain on this because the gram stain isn't going to work because it's not going to, the stain's not going to get the cell with alcoholic acid. So you have to do an acid-fast stain. The way the acid-fast stain works is you do the fixation and everything the same way. But when you put them on the slide, instead of adding crystal violet for the gram stain, you add this primary stain. It is called basic fuchsia. You'll see it also called carbon fuchsia. And you add that, you flood the slide with that over a steam bath. So the steam heats that slide, and as it heats the bacterial cells, it allows this basic fusion to actually penetrate inside the bacterial cells. So at this point, whether you had things that make mycolic acid or not, everything's going to be this color, like a red color. Then you add a decolorization step with acid alcohol, so a little bit more than just ethanol. It has an acid in it as well. That's going to be your decolorization step. The mycolic acid, when you remove it from the heat, is going to, I'm trying to think how to explain this. Like you light a candle, and it becomes liquid when it's hot, right? You take it, you blow out the flame, and everything solidifies again. It's kind of what's happening here. You remove the heat, the mycolic acid kind of recongeals, and it'll trap this inside. If it's a bacteria that doesn't make mycolic acid, this acid alcohol will decolorize it. So technically in the acid-fast stain, this is your decolorization step. It's like, well, I know I put something on the slide, I don't see anything anymore. That's because it's not making mycolic acid, it's something else. Probably some kind of a gram bacteria, gram positive, gram negative. So excuse me, decolorize without acid alcohol. Down here in your counter stain, to counter stain the things that don't make mycolic acid, you can use methylene blue. Methylene blue is going to stain the bacterial cells that don't make mycolic acid blue. They can't get, the methylene blue can't get inside the ones that does make mycolic acid because you removed it from the heat at this point. Or sometimes some of my older colleagues will use malachite green. It's just a way of, I can't remember what year that changed, but one of my old bosses, this is a entire long time ago, always used malachite green. I don't know. Essentially, you use a stain that's not red. Yeah, that's all it is. Yeah.
Is this only for gram negatives?
What do you mean?
Like,
That?
once you figure out, like, from a gram stain, if it's gram negative, will you do an acid-fast stain?
No, so acid-fast bacteria are completely different. You have gram-positive bacteria, you have gram-negative bacteria, or you have organisms that make mycolic acid. And the mycolic acids are like mycobacterium, tuberculosis, nocardia, and those are, they're different. They're very different. And what you'll get, the reason you do the counter stain, especially if you take like a respiratory specimen, sometimes you'll get mycolic acid reducing bacteria in there, like mycobacterium, say they have tuberculosis. But you might get some stuff in the respiratory tract too. You just might have some regular run-of-the-mill that might be gram-positive, gram-negative, this might be something else in there. So you might see a mix. This is a really good stain. You pretty much only see the TB, but usually you might catch something else too. It's just so you can see. This is what the structure looks like when mycolic acids are really thick. So that's your acid-fast stain. Outer membrane, let's take 5 actually. Let's take 5. Let's take 5. I feel like this semester is aging me rapidly. Let's take 5 and we'll come back and finish this up. And we don't have much to do for labs, that's a good place for last time. Oh, they're good. They're good.